Okay, sounds good. Thank you, Sam. So it's my pleasure to introduce Adriana Comprelli. She is a executive director of therapeutic area head in clinical operations at BMS. She is responsible for the operational strategy and planning for a portfolio of innovative assets in early clinical development. Ms. Comprelli has over 24 years of sponsor-based drug development experience. She began her career as a project manager at a CRO focused on inhaled insulin and immunology programs, and later accepted a position at Aventis, which is a global pharmaceutical company, as a medical research associate in immunology. Adriana joined BMS in 2003 as a clinical scientist supporting registrational programs, and she continued her contributions across the oncology and immuno-oncology portfolios. Currently, she manages numerous programs across a broad portfolio, including adaptive and innovative early development oncology trials, as well as immunology, cardiovascular, fibrosis, and neuroscience programs. She also heads the operational strategy for clinical pharmacology in all phases of development and therapeutic areas. She holds a BA from Rutgers in psychopharmacology and a project management certificate. Her contributions have resulted in numerous publications.
Thank you. I will share my screen. Good afternoon, everyone. So I'm so excited to be present with you today. My introduction was so kind. Thank you so much. I'm really excited to talk about the innovative and operational aspects of clinical trial management and portfolio management, specifically in early phases. Typically in phase 1A, we don't have room for operational innovation or new ways of working, but I'm going to share some methods and case studies where we've applied some tactics to drive truly industry-leading metrics. And I'm really excited to share these tactics with you. When I was preparing for this talk, I shared with my boss, my mentor, and they had given me feedback that, typically in these presentations and sessions, there's nothing tangible that's given. There's nothing that you can take away and really action and bring back to your organization. And surely that has a lot to do with the industry that we're in. But I'm excited to share things that we've been working with academic centers and community centers across the board that really have become more public. And I think they can really spur some innovation in clinical trial asset development. For innovation, I really do believe that it's important to think about where you have capacity for risk in your function and for your organization, but for as the business as a whole. So we'll talk a little bit about that as well, and talk about some of the asset journey in terms of clinical operations. Let me see, I am not presenting in full screen. Just as a conflict of interest disclosure, I am a BMS employee and stockholder, and the views expressed in this presentation are my own. I wanted to focus on a few aspects of clinical trial innovation in early phase, specifically in site collaboration, and share some of the really interesting case studies that were completed in early asset development. Also, some lessons for regulatory considerations and operational impact. And then how we've used some what are typically late phase or full development collaborations through our business and modeled them into early phase that really drove some amazing metrics and collaborations. I'm not going to talk about operations and protocol design and data needs or some unique operational processes that were developed, specific SOPs and processes that were developed. But they are important factors to the whole of this story. Also, I wanted to share that the framework for this will be truly patient-centered advanced cancer clinical trials through phase 1A and phase 2A. So really novel first-in-human studies, and that's the focus of my career right now. And within those, we found that there are true acceleration opportunities in early development. And I know we've all seen how the study paradigm has been remodeled in the past couple of years, specifically in the immuno-oncology space and oncology space with a lot of our monoclonal antibodies. So the speed of early development in the entirety of the footprint has really, really changed in the past 5 to 10 years, and specifically in the past few years. But some of the acceleration opportunities that we took upon were identifying unmet medical needs, looking at an expanded site and country footprint specifically for phase 1A, which is a very unique site need, and more especially in immuno-oncology and oncology. Some really great novel regulatory approaches that comes with the development of new molecular entities, and fast paths to registration, something that's been really exciting in the past few years. Also, patient selection and adaptive design, and we've had some great presentations on these topics today at this session. Some of the challenges we faced is the FDA and health authority approval on novel dosing strategies and formulation strategies. And how do you combine that in novel study designs and balance safety as your primary endpoint with an early picture for efficacy, if possible? Also within those novel dosing strategies, what the patient flow looks like, getting data early on IV and then moving to something like subcutaneous or oral within the same study or program. It's important that we understood the limits and opportunity of the patient populations in context of very multiple layers in large and broad first-in-human trials. And working in country selection and site selection where you had very limited or no human data at all executing first-in-human trials. And then, of course, something that impacts many of us is biomarker handling. And operations is really key in the fast and timely and quality turnaround of biomarkers. And in small sample sizes, that's so impactful to early development trials. I'm really excited to share some of the results first of the activities that we've done. So as you can see, this is in regards to very complex first-in-human oncology patient trials in a global setting, so not just US-based. I also like to share that these are in-house, in-source trials. These were not outsourced trials, and that's an important factor in this. I do believe that you can get to working with a vendor, a CRO, that you can fully outsource and still achieve this level of metrics, but it really is dependent upon a very high touch with that CRO and very, very well defined expectations. But within the past 3 to 5 years, I've been able to move 24 assets into clinic from preclinical, and some of the timelines that we've achieved in the US and ex-US are listed here. These are all in the public domain. Our greatest achievement was 1.6 months that included an IND submission, IND and protocol amendment, and site activation. The average from go to first-in-human to first-in-human dose that we're achieving is about 8.5 months in these complex oncology trials. And protocol approval to first patient in is about 39 days average actual. So I'm going to share with you how some of these metrics were achieved. I want to talk about the type of studies that we're doing. So as I mentioned earlier, the development paradigm has absolutely changed. We no longer have very simple first-in-human SAD/MAD studies in a complex population space. Larger sponsors and even smaller biotechs are really looking to get the most from their early development pathway and their early development patients that they're enrolling, if they're in inpatient trials, and look at the model of first-in-human to potential peri-registrational or registrational trials within the same study. The FDA has certainly wavered and moved back and forth around the ability to do this, and I found that it's really dependent upon the molecule itself, the population. But in totality, for the past several years, we've been really working in this paradigm where you have your phase 1A and signal all the way through signal expansion based into the same trial. So most of the first-in-humans I've worked on are upwards of 600 patients. That bring us to pre-registration or potential peri-registration opportunity. As we saw in an earlier presentation, a lot of this is dependent upon novel statistical design. The allowance of cohort backfills is really important to get more data in a smaller sample size. And I've also worked in the majority of trials where we not only interrogate monotherapy, but also combination therapy. And that can be with 2 novel molecules in combination dose. So I'll talk about that a little bit as well, and also those novel dosing mechanisms. Moving from that, there's signal exploration. The FDA has really charged us with limited opportunity for sample size and signal exploration when we have a non-specific IND. But this allows us to come to our recommended phase 2 dose, and then we go to signal expansion. And I've listed here my typical timings and cohort sizing. And that signal expansion is typically the opportunity to move to registration. So now the goods. So site activation. I want to share with you what we brought to our clinical centers that we partnered with, and I'm going to talk about those collaborations. But in site selection, we've developed a process that's really nuanced to early asset development and those larger phase 1A studies. What was important is the development of an FTE that really specialized in portfolio planning. BMS in and of itself had about 32 molecules across 2 to 4 years ready for first-in-human development. So this pre-first-in-human role really was someone who looked at it at a portfolio level and demonstrated that you can have site collaborations, early planning, to really have a nice machine of execution and operations. The big picture is we would move very, very selectively in dose escalation. We heard from sites that having more than 3 to 5 sites in a dose escalation was inefficient because everyone's fighting for slots, as you may know, et cetera. So we really looked to commit to our site that we would only add 3 to 5 centers for this phase, but they had to commit back to us that they would prioritize and enroll our trials. And that commitment was mutual, and it worked beautifully. We looked for centers that had a specific MOA match. Oftentimes they had experience with competitor molecules or even in their own academic centers, but it was important that they had some level of understanding of the mechanism of action or the molecule itself. And it was imperative that they had really strong translational capabilities. I will talk about later in the presentation site assessments that were specific to this model that were really successful and drove the collaborations. And through these collaborations, I was able to achieve this speed from IND of 4 to 6 weeks. My best time was with a site in Canada, a very popular, famous center, where they achieved health authority approval to first patient dosed in 5 weeks. And it was using our collaboration model. So it was really, really a wonderful experience and drove other centers to try to match that. They all asked, how do we get to that? So we brought to them this program to say, this is how we'll model with you. Then if you look at signal expansion, it's typically 10 centers. Those initial 3 to 5 centers act as your KOLs. They open up to a deeper patient population. The sites are assessed as phase 1B sites, so a little bit more relaxed in terms of MOA experience, but still needing that very strong translational capability. But we did look for leadership, indication leadership from all different sources, including our medical group, publications, etc. And then the phase 2 expansion, we really treated as a phase 3. So preparing for the global footprint, we looked at thought leader mapping early on and really tried to source against the patient population. So that was a really important piece to not look at these as expanded phase 1 units, but really as potential phase 3 centers. This is a little bit about the selection and timing. So what was really important feedback from our centers is that they participated with protocol development if they were in that first in human phase with us. And often if we had sites that were interested and they couldn't get in as those initial 3 to 5 centers, they were very interested in participating in one of the escalation phases, that may be the phase 1B escalation. And then the phase 2 sites, we brought in 4 to 6 months prior to expansion to give them enough time to train on the protocol, align with their health authorities, and also prepare if there were any amendments with outcoming data. So our best case scenario for these are listed on the slide, and as time went on, we really were able to become more efficient as we built collaborations with our centers. Of note, we're up to over 100 centers in the US that work in this model with us, and have a very robust global footprint as well. So this is what we actually work with our centers on, and I have some very interesting case studies. So as you can see, we come to them with the goal of 4 weeks from IND approval to have their first patient screened. We come to them with our portfolio, so there's very strong alignment and there's the promise of a collaboration and connection, but we also work with their on-the-ground operational and regulatory staff to really drive this turnaround time. Many centers that weren't doing parallel reviews and approvals or things at risk agreed to work with us at risk. So, sometimes you just have to ask, you have to inform them and say these other sites are able to accomplish it. I feel like we have more leeway in the phase 1 space. Phase 1 academic units are typically more interested and able to move through these review bodies a little bit faster because they are more niche. So they agreed that all of this is done at risk. We may have very strong feedback from the FDA, but we're able to come to this. Some of the centers do ask for pay for play. So if we want to do this, it is an upcharge because they're aligning additional resources and time. One of our greatest successes, there's a very well-known academic center in New York where they were building their early development unit and said, what can we do? And they were stuck on the idea of a local IRB, which was incredibly rate limiting. It was several months long. I shared with them that the NCI had a central IRB initiative, and that's something you can find on the NCI website, where they were allowing central IRB review for the main protocol, and then a smaller local IRB review and local IRB management. They brought that to their IRB review board and did approve for early development studies that they were able to use a central IRB. And that really, really changed the paradigm for them and our ability to use them as one of our first in human centers. Another center that's a well-known academic in Michigan were just building their phase 1 unit. And it's not a normal healthy unit, it's a patient unit, and really wanted to know, can we partner with them to bring in novel operational processes? So we sat one-on-one with a lot of their team members and very granularly went through opportunity and process, and I can say that they've really built a nice unit for early development. These are just some of the pieces that we bring to our centers to display what makes it a great phase 1 center and someone that we want to work with. The most important piece is the investigator has to be engaged and has to be ready for one-on-one calls. They have to be willing to share their cell phone. They have to take full accountability for these studies. The smaller sample size, the intense activity with a very sick patient population requires that, and we need a strong representative. So that's really one of the most important pieces. And then a lot of the laboratory staff, dedicated staff typically is the best option, but as we know, a lot of centers seem to share staff, and we can work around that. I'll talk a little bit about the hub and spoke model, but I think most people would be familiar with that. But really creating a relationship with us in our early development group was one of the most important factors for them being strong collaborators. And I highly recommend that your operational team have opportunity to create those relationships post-COVID, be on site, and really get to know the staff. That makes for the best practice for collaboration. The hub and spoke model we've adapted into a continuous partner model framework, where we really give these units the opportunity to holistically have the program from early development to registration, as long as they continue that commitment with us. So we'll start with a small isolated dose expansion or a first in human. We'll ask them to really be leads on our program, help us evaluate for indications, look at combination therapies, but really be fast in their ability to share their data with us, get that data in-house, and do almost real-time data translation with us. And that's an imperative for these centers. And then from that, we pivot to signal opportunity and registrational opportunity. And they self-identify indication leads to support that late phase enrollment. And it really becomes an asset journey for the center as well. So it's worked a little bit deeper than a typical hub and spoke model. And many of our centers are very interested in that. I think one of the caveats to this for BMS has been we have a robust pipeline that keeps them interested and engaged. So it's important to find those ways in with centers to increase their interest if you have a smaller pipeline. I'm going to talk a little bit about some of the health authority lessons that we've had in case studies. Maybe we all know they're broad ranging and not aligned by country, so that can be difficult, especially in an early asset development program. It's really based on the molecule mechanism of action. Some examples with our agonist molecules, much harder to get through health authorities without data. Also what experience that country has in class. And even to the point of legality within their country laws regarding adaptive design. That was an interesting lesson. So for flexible language, it's certainly gotten worse over the years in terms of the allowance of flexible language. And I think this is a place where it demonstrated to us that operations really needs to be at the table as part of the development team. They can't be a subgroup that really just executes. They really have to be allowed to be at the table and talk through potential options based on their experience. So when we've looked for flexible language in some of our protocols where it would be based on incoming data or unknown data, we would do this and maybe we would do this and not really spell out the phase two. The health authority said that lack of clarity is not something that they want in their country. Sometimes when they did allow some flexible language, it was only because we said, we will do this if the data shows this, and was very specific on what that phase two would look like. But typically, the health authorities did not like flexible wording, flexible protocol language. They found it complicated, the review. There was confusion within the trial elements also if we did execute that and then had to go back with large amendments, and used instead planned amendments. So we showed them what the amendment would look like, and that was much more successful. In terms of protocol complexity, most health authorities, when we tried to jam in numerous exploratory arms or different arms and really make a large study out of a first in human, the health authorities said that this was really too complex. It was 6 protocols in one. And they really wanted us to focus on what were the priority elements of the initial submission. And then when more data came in, look at additional elements, perhaps amendments that would support the new endpoints or other endpoints that we were interested in. Impactful to first in human is, of course, the starting dose. And in novel biologics, it really can be differentiated based on its MOA. So we also found that the health authorities tended to be really conservative regarding the approach that they wanted in terms of first in human dosing. They wanted a sentinel patient for every dose level in some of our agents. And if we had pre-planned that, it would have allotted for a faster health authority approval. Often they look for a robust preclinical data set, but it's interesting with novel combinations. When it's 2 novel assets, they do not require the combination preclinical data for those novel combinations. You can rely on the individual asset data for initiating combination studies. So that was a great thing, and I think great progress in the FDA and health authorities. In terms of initiation of those combination arms, often the starting dose has been challenged. If you don't have combination preclinical data, they really prefer that you start at lower doses. Often for our antibodies, they have accept pharmacology-based rationale, and that's been very successful. So we really bolstered our pharmacology programs. but they all required that the monotherapy data be available at the time of the initiation of the combination. So you can have almost concurrent monotherapy and combination therapy arms in dose escalation, but they do need a subset of monotherapy data and safety data submitted prior to the initiation of the novel combination. And that's certainly something that has expedited our development plans for early assets. So in summary, in particular for these IO agents or novel agents, there's high scrutiny. Flexible language and flexible protocols have actually made it harder for us when we thought that having some flexibility and open-ended wording would make it easier on sites and on us in terms of amendments. It made it harder with regulatory reviews. And then for all MOAs, the starting dose, how long the DLT period was, and the sentinel patient were all key elements of health authority reviews. So really focusing and having strong rationale and basis for your recommendations and what you're putting in the protocol prior to IND review is imperative for your health authority review. I want to talk now about our strategic collaborations, and these were some of the things that really drove those high quality metrics that I shared. 10 years ago, we had a few US centers that were eligible to do first-in-human patient trials and very little global centers. Within that time, and more specifically in the past 3 to 5 years, I put in a process where we went to academic centers and community centers, which we'll talk about in a moment, and went through not what's a due diligence, but a phase 1 site assessment that was really a feedback loop. We worked with the center, we selected high-performing academic centers or centers that were new and trying to build their phase 1 units, and spent, it could be 2 days, but many hours on site with them. The purpose was to qualify them based on their ability to execute a first-in-human trial and look at all of the operational and clinical aspects of their work. I've been to some centers where one nurse is in charge of dosing 6 first-in-human patients in different rooms on the same day, and there's no physician on site. Or other ones where they take it much more carefully and the physician doses and it's several hours and they'll limit it to 3 first-in-human patients for different protocols in one day. So you really have to understand what is your comfort and what is important in terms of your molecules and their safety. But what this brought was a large collaboration. They enjoyed seeing our portfolio. They enjoyed learning how the team works and would help them get to participate in first-in-human dosing, or what they needed if they were only really set up for phase 2 delivery. What did they need to do to get to that first-in-human delivery? As a note, our legal team said we could not call these due diligence visits, and they were much more intensive than pre-study evaluations and typically included executive leadership as well. So that really brought to these centers a strong sense that we wanted to partner with them and help them grow to help us grow the portfolio. One of the novel things we did was access our community networks. There was a very strong call from some of the US community network groups that they wanted to get engaged into phase 1 development, but those community physicians typically didn't have the site capabilities or the expertise to work in a phase 1 setting. What we did do is ask a lot of the networks that were interested in this to self-identify who in their community sites were capable, where they partnered with a local academic hospital to potentially do this. And we executed the same type of assessments. And I could tell you from about 20 centers that we visited, 7 were qualified as phase 1B, and 1 was qualified as a phase 1A. The ultimate goal of this, which was really successful, is building a continuum of care. So allowing our physicians in the community to get early access and their patients to get early access to some really novel potential treatments for their patients, but have that early experience moving into full development and approval and post-approval. Typically, these community physicians are not experienced with agents when they come out. They stick to the tried and true, and patients have to travel to academic centers. So it was really important to us to look at this from the patient's perspective and get these novel molecules and experience to their community physicians as a service to our patients as well. I can tell you that in phase 1, it was very limited success. They could not manage the contact that we needed, the really detailed contact points and action points. So right now it's typically best for early phase 2, but it still works in terms of getting an expanded patient population, enrollment options, but getting these communities ready for some really important treatments. Also, one of the novel frameworks we did is with our medical affairs group. So typically only full development or phase 3 is assigned field medical science liaisons to support a lot of the activities at the centers. We partnered with them to formalize a framework and develop a very specific phase 1 qualification questionnaire, and also molecule information for our field medical teams. So if they're at these centers that they feel may have a potential contribution to the early asset development process, they can screen them and see if they're a good fit. So the addition of having those additional field personnel was a really big leap for us, and they felt that they can speak wholeheartedly to the entirety of the company's portfolio, what's to come, and potential collaborations. A specialized service, which was really great, is our field medical team also has pharmacodiagnostic support. So typically these are pharmacists. It was such a great thing for early development. So when we had these novel assays to process really delicate samples, we can rely on these field agents to go to the centers and do on-site training and on-site support throughout the course of the trial, where typically we wouldn't have that. So that was a really successful option for us. And actually, that's what this is, sorry. So in summary, looking to expedite operations for early development, build out novel activation processes, look at portfolio collaborations, conduct some in-depth phase 1 site assessments. As a sponsor, if you're working with a CRO, don't rely on your CRO. You have to build that one-to-one connection with them. Have specialized roles if you have the FTE space for that, or assign someone to first-in-human planning and delivery. And make sure you embed phase 1 needs holistically through your field force as well. We found regulatory agency roadshows were imperative in our success for an early development portfolio, and that was global. Updating the hub and spoke model to a more in-depth continuous partner model in a center was highly successful. Using novel strategies, as I mentioned specifically in Canada, really supported our metrics and outputs. And something I didn't talk about, but look at your processes and don't rely on phase 3 SOPs and processes in your business to drive early development. They really need to be differentiated for early development, so you are allowed to move faster and not do process for the sake of process, but really manage novel ways in early development. Okay, thank you so much. I'm going to exit.
Hey, Adriana, that was terrific. Thank you very much. And I can attest to personal experience collaborating with BMS that the clinical trials operations team there is just incredibly impressive and phenomenal. Just how quickly and collaborative your teams are in terms of getting trials up and running. So it's really a terrific group that works there. Now, one thing that you touched on was that BMS has a really nice large pipeline, is able to have that out as a carrot to really get investigators and sites engaged. Do you have any suggestions for smaller biotechs who may have maybe 1 or 2 assets in the clinic to try and take advantage of some of these some of these strategies?
Absolutely. Going out to these centers and speaking to these physician researchers, they want to be a part of it. They want to be deeply involved, and they unfortunately don't want the initial connection to be from a CRO. The clinician really should, the sponsor clinician really should connect with the academic clinician and get their input, have them as a part of protocol development, have a small consulting agreement. The more involved you bring them in, the more committed they'll be. Many of them ask for publication rates, and I found that you don't need to promise that. But you should promise collaboration and potential future opportunity, leadership opportunity, and that seems to work really well. They want that one-on-one sponsor connection most of all.
Yeah, I can completely agree with you, especially being involved early in the protocol writing. A lot of times big pharma, just shows up with a protocol and says, "Here, we want you to do this study," and that just doesn't fly with a lot of folks. They just don't, have much interest.
Exactly.
Yep. All right. Well, thank you. This was really terrific. I enjoyed it.
Thank you.
Again, thank you, Adriana, for your presentation. And yeah, if you guys have any questions for her, even after the event, let us know. We can email our questions to her. And without any further ado, I will be transferring everyone to the next presentation.